with both of the sites available for the mammary gland are lactating. Okay. There is a portion of this slide showing us the almost regular appearance of the histology of the mammary gland. Now, mammary gland is a compound acinal gland. It's a compound acinal gland. The secretory portions are called the acini. The point, they are the acini. Now, just like in the parotid gland, which is also a compound acinal gland, the acini are grouped into lobules. So that is one lobule. Can you see the pointer? Okay. That is one lobule. That is another lobule also. So any duct within a lobule is called intralobular duct. And then that duct which is outside of the lobule, intra or interlobular? Interlobular duct. Now, so shown now is one lobule of acini. One lobule of acini. Okay? Now, the acini, being the secretory portions, are lined by simple columnar epithelium. Fortunately, the stain is very faint already. Okay, so that is one acinus, another acinus within a lobule. So those are the columnar cells comprising the secretory cells. Now, surrounding the secretory cells are those cells which contain actin and myosin filaments. And because they contain actin and myosin filaments, they are called the myoepithelial cells. The myoepithelial cells. What is their significance? Okay, they contract in order to eject, squeeze out milk during feeding. Okay? Now, it is an interlobular duct outside of a lobule. So, usually the ducts are lined by what type of epithelium? Simple cuboidal. Simple cuboidal epithelium. That's the simple cuboidal epithelium. It is also surrounded by flat cells. They are also surrounded by myoepithelial cells. So the myoepithelial cells are present surrounding the acini and the ducts, intra and interlobular ducts. Now, that is the histologic appearance of an inactive lobule of acini of the mammary gland. The rest of the field, the rest of the tissue, will show us lobules of acini which are actively producing milk. So look at the increased number of acini per lobule. And then look at also the increase in the size of the individual acinus. So compare that to the lobule which we have previously studied. Ang lobule na yan. Check yung pointer. On the right upper portion, that was the acini at the lobule that we have studied a while ago. Okay, and then the interlobular duct, and then look at the remaining, the other lobules, the ones that are pointed right now. Ayan sila. Okay, look at the difference in the size of those acini and the size of those acini. And then the marked crowding in the left portion. Marked crowding of acini in this shown area. Ayan. So, the acini are larger than the usual, and then their lumen is dilated. That pointed material is actually milk.
Yung pink. Sa gitna. Sa gitna. Ayan, last name.
May laman.
Okay. So this is one asinus, another asinus, and then another asinus. Notice the dilatation of the lumen of the acini to accommodate the secreted milk. Ito yung milk, ito yung secretory vacuoles at the apical portion of the secretory cells. Now, what is the color of the milk that you can see? Pink. Baka strawberry flavor yan. That is the appearance of a lactating mammary gland. Lactating mammary gland. That is the appearance of an active lobule of acini. And then this portion will show us a lactating portion of the mammary gland. So the ducts are also accumulating milk.
Kapag may color ng loob,
Like my pink.
Same
yung
level
loob,
content is
active.
Active.
on the mammary gland. Lactating mammary gland.
Nice.
Ito, hindi masyado. Itong tatlong lobules na ito, hindi masyado increase ng kanilang secretory activity. In comparison with the other lobules, which are exhibiting increased number of acini. Now, for the histology of the organs, including the male reproductive system, we'll begin with the histology of the testes. The testis is covered by a dense irregular connective tissue, which is called
tunica
albuginea. Immediately underneath the tunica albuginea is a connective tissue which contains numerous blood vessels. And because of the presence of the blood vessels, that layer is called tunica vascularis. Tunica vascularis. Now, the functional units of the testes are the seminiferous tubules. And the seminiferous tubules are seen most of the time in the cross-sectional views. Sila. So actually, in reality, seminiferous tubules is a continuous tube which is highly coiled. Kaya, most of the time, we will not be able to see the longitudinal view of the tubules, but their cross-sectional view only. If we stretch out the seminiferous tubules, what is the length that is indicated in the book?
50 cm lang.
Cm now? Is why, what should be the average number in millions of spermatocytes per ml? 7. 3 million lang?
8 million.
Okay. Now, the seminiferous tubules are responsible for the production of the sperm cells. Okay? Collectively, those cells inside the tubules are called spermatogenic cells. Spermatogenic cells. Now, among the spermatogenic cells, the most primitive are called spermatogonia or the spermatogonium, singular form. Okay, how will you be able to identify the spermatogonia?
Near basal lang.
Okay, so they are very easy to identify because of their characteristic locations. All you need to do is to locate the basal lamina of the tubule, and then any cell that is immediately adjacent to the basal lamina is called
Spermatogonia.
Okay, and then following the spermatogonium is what stage of development?
Secondary.
Okay, spermatocytes. Now, the Spermatocytes are very easy to identify also because of their size. Okay? So the cells pointed right now are spermatogonia, very near the basement membrane. The largest among the spermatogenic cells are
Spermatocyte.
okay, primary. There are two types of spermatocytes.
Primary.
So those cells which are much larger than the other cells inside the tubules are called
Primary spermatocyte.
primary spermatocytes. Pinakamalaki sila sa spermatogenic cells. So primary spermatocytes, that layer pointed. And then immediately above the primary spermatocytes are,
Second.
you will not be able to see the secondary spermatocytes in the seminiferous tubules. Why? Before they mature, the primary spermatocyte will have to pass through the secondary spermatocyte and become spermatid before maturing eventually into a spermatocyte. Why will you not be able to see the secondary spermatocytes? Because they immediately enter into the second meiotic division to produce the cells above the primary spermatocytes are called spermatids. Spermatids initially are still round cells. How do you call the process of transformation from round cells to eventually cells that will possess mid-portion and the flagellum? How do you call that process?
Capacitation.
So from that layer, okay, identify the cell pointed. Okay, based on the size of the nucleus primary, how about that cell? Spermatid. Identify the cell. Okay. Identify the cell pointed. Spermatogonium. How about this cell? This cell.
Primary.
Spermatocyte.
Primary.
Primary spermatocytes. How about that cell?
Spermatid.
Spermatid. How about these cells with elongated nuclei?
Sertoli.
The Sertoli cells are actually the largest cells inside.
Lidig sa labas.
The Leydig cells are located outside of the tubules. These are the Leydig cells. They are still the spermatids. How will you be able to differentiate a tailed spermatid from a mature spermatocyte? Say the last stages of development of the spermatids, they will already possess the midpiece and the flagellum. How will you be able to know that a flagellated cell is still a spermatid and not yet a mature spermatocyte? Masyadong makitang Sertoli cells. anyway, remember that the shape of the septolyzed cell is pyramidal. And then inside the seminiferous tubule, the largest is the septolyzed cell. Pero among the spermatogenic cells, the largest is
Primary.
the primary spermatocyte. Okay. Identify the cell pointed. How about these cells? Spermatids. How about these cells? Spermatids. How about these cells? Okay, the spermatocytes, the mature spermatozoa. So mature spermatozoa. You will be able to say that a flagellated cell is already mature if it is already detached from the germinal epithelium. Any flagellated cell that is still attached to the germinal epithelium is called?
Spermatid.
Is called?
Spermatid.
Any flagellated cell that is attached to the germinal epithelium should be called?
Spermatid.
Spermatid. Spermatid. And then how about these cells? With abundant pink cytoplasm outside of the seminiferous tubule. What hormone is produced by the Leydig cell?
Testosterone. Testosterone.
Testosterone. And then the Leydig cells are stimulated by what pituitary hormone to produce the the hormone testosterone. LH or there are two types of gonadal proteins.
LH saka FSH.
So that is a group of what cells? Interstitial cells of Leydig. Outside of the tubules. Okay. And then those spindle-shaped cells are, aside from the Leydig cells, what is the other type of cell that is present in the interstitium of the testis?
Myoid.
On the myoid are located immediately surrounding the basal lamina of the seminiferous tubules. They will help squeeze out sperm cells during ejaculation. So, a cell with pink abundant cytoplasm outside of the tubules is called based on the appearance of the interstitium, what type of connective tissue is composing the interstitium? Loose connective tissue. And then these cells with abundant pink cytoplasm are called? They are called? Okay, interstitial cells of Leydig. Now, notice that there is another type of cell present in the interstitium. These are the cells responsible for producing the collagen fibers in the loose connective tissue of the interstitium. This cell should be called? Very good.
Fibroblast.
Following the seminiferous tubules, what is the first duct that will serve as the passageway for the mature spermatozoa?
Intratesticular ba?
What?
What's the first?
Intratesticular.
Before entering the epididymis, there will be a series.
Intratesticular. Extra.
epididymis. Just like the seminiferous tubules, the epididymis is also a highly coiled tubular organ. That is why we will be able to see cross-sectional views of the segments of the epididymis. Now, the epididymis is lined by what type of epithelium?
Pseudostratified columnar. Pseudostratified columnar.
Complex
Okay, what is the shape of the cells lining? Pseudostratified columnar. And then how do you call those structures projecting from the surface of epithelial cells?
Stereocilia.
What's the significance for the presence of the stereocilia?
Movement.
Are they motile?
No.
Are stereocilia motile? No.
No.
What is their function? Stereocilia are actually elongated microvilli. What is the function of the microvilli? Increase surface area for absorption. What is absorb? Okay, to make the semen concentrated, those structures are present to absorb water from the semen and then identify the cells pointed. Spermatozoa. Low power magnification, that is the appearance of the epididymis. On scanning objective view, that should be the epididymis. And then this tissue here should come from seminiferous tubules or the testes.
Oh, the column.
From blood.
This, and then immediately adjacent is the epididymis. Following the epididymis is the vas deferens. Okay. Now, are you familiar with vasectomy? Okay, that is the organ that is being tied and cut in vasectomy, the vas deferens. That is why it is called bisectomy. Okay. The vas deferens is the most is a mascular tubular organ. Okay, what is the lining epithelium of the vas deferens? Okay, still pseudostratified columnar epithelium. And then, projecting from the surface of the epithelial cells are yan, yung parang mga buhok-buhok. Stereocilia. And then, the cells inside the lumen are called spermatozoa. Okay. How many layers of smooth muscles are there in the wall of the vas deferens?
3.
Okay, 3. And then the outermost layer should be called?
Adventitia.
Are you able to see a smooth surface? No. So that is the external surface, or the outermost layer should be called?
Adventitia.
Okay. Vas deferens. Okay. We're now looking at the histology of the penis. What is responsible for the erection of the penis?
Erectile cavernous bodies.
Para.
Okay. The
Vascular.
feeling of blood, oh? Cavernous bodies.
Cavernous.
Now, the cavernous bodies are actually composed of what? Structures. How many cavernous bodies are there?
3.
3. Two dorsally located, which are called?
Cavernoso.
Cola cavernosa and
Spongioso.
almost spongiosum. Now, the very easy to identify. That portion shown is the corpus spongosum. Okay? Now, the cavernous bodies are composed of veins. Those spaces are actually veins. During erection, these veins will become filled with great volume of blood. Kaya nasa dugo lang yan. Okay? So, these veins will become filled with blood, and then this is the lumen of the urethra. Okay? The urethra may be lined by different types of epithelium. Near the urethra and near the urinary bladder, the urethra will still be lined with transitional. Okay? In the membranous portion of the urethra, it will be lined by stratified columnar. Near the urethral orifice, it will be lined by this type of epithelium, which is stratified squamous epithelium. It is very easy to identify the corpus spongiosum because of the presence of the numerous blood vessels with the centrally located urethra. This is the urethra. Now there is a gland associated with the urethra. Makita dyan.
Urethral gland.
Anong tawag sa gland na yan?
Urethral gland. Urethral gland.
Bulbourethral gland also opens to the urethra, and then the smaller mucosecreting gland, the glands of, the name starts with letter L, Letrae. Okay? Now for the males, if you have noticed, when you become sexually stimulated, you achieve erection, may manotice kayo maglabas ng mucus from the urethra. So those mucus secretions are coming from the bulbourethral glands and the glands of Letrae. Now the physiologic significance of the mucus secretion is to lubricate the vagina during sexual intercourse. Kasi painful sa mga babae ang dry sexual intercourse. Kaya mayroong mucus secretion from the glands of Letrae and from the bulbourethral glands. So that is the corpus spongiosum and then the boundaries of the corpus spongiosum. So the corpora cavernosa will still be composed of numerous venous channels. However, they are easily distinguished from the corpus spongiosum because of the absence of the urethra. And then, the penis is covered externally by what type of epithelium?
Stratified squamous.
Stratified.
Squamous.
Kulang pa.
Minimal keratinized.
Keratinized.
Minimal.
Okay, that's the keratin layer. How about the vagina? What is the epithelium lining the vagina?
Stratified squamous. Non-keratinizing.
Non-keratinizing. You can just imagine kung keratinizing din ang vagina. Kung sexual intercourse, mag-aso-aso. And how do you call the connective tissue immediately underneath?
Lamina.
How about those smooth muscles? Immediately underneath the loose connective tissue.
Muscularis.
Anong specific name yan? That will be your assignment.
Ano specific?
And then how about that connective tissue immediately surrounding the cavernous bodies? Mayroon din specific name yan. That will be your assignment. Nakalimutan ko. Okay? Corpus spongiosum is characterized by the presence of numerous venous channels with a centrally located
Urethra.
And then corpora cavernosa is characterized characterized by the absence of urine.
Urine.
And then that artery, which is located at the dorsal of the penis, is called
Deep dorsal artery.
okay, deep dorsal artery. What is the function of that artery? To supply blood to the cavernous blood vessels or to supply nutrients?
Okay.
Which artery or what artery will supply blood to the cavernous blood vessels during erection? And it will be in the parlance of the lecture already that the officers.
Helicine arteries. Helicine arteries. Helicine arteries. Increases the blood flow.
Okay, for the next organ, prostate gland. And center. Now, just like the mammary gland, the prostate gland is a compound sinus gland. So being such types of gland, it will be composed of the sini, which are grouped into lobules. So this is one lobule of acini, another lobule of acini, and then another lobule of acini. So the lobules are separated from each other by a specific type, distinct type of connective tissue or stroma. What is the specific name of this stroma? Being composed of a highly fibrous and muscular components. You have the acini, the secretory portion, and the fibromuscular stroma because of the presence of numerous smooth muscles. Now, the smooth muscles during ejaculation will also contract. So to squeeze out the secretions of the prostate gland. What sugar? Fructose is coming from what gland? Seminal vesicle. Okay, what is the epithelium lining the acini of the prostate gland? Stratified or simple?
Simple.
What is the shape of the epithelial cells? Columnar, simple columnar epithelium. There is also the myoepithelial cell layer, immediately surrounding the simple columnar epithelium. Simple columnar epithelium, the myoepithelial cells. Myoepithelial cells. Simple columnar epithelium.
Sorry.
Okay, for the histology of the seminal vesicle, all you need to do is to memorize that appearance.
Bilog-bilog.
Okay? So that is the histology of the seminal vesicle. The secretory portions are lined by what type of epithelium? Epithelial, those epithelial cells. Okay, what type of epithelium? And then, what type of tissue is surrounding the secretory portions?
Close.
Yan tissue niya.
Close connect.
Anyway, I will not be asking that question. What you need to do is to memorize that appearance on the histology of seminal vesicle, and then what sugar is present in its secretion.
Fructose.
Do you have any question? We will have a quiz